Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all, and welcome, and thank you, SynBYSS people, for joining us, the 133rd seminar. I call it a Northwestern Day. I came back to St. Louis after my Boston trip and giving 2 talks at an MIT energy conference and Draper laboratory near MIT, exchanging great ideas and discussing synthetic biology and its solution to global issues. I also met 3 SynBYSS young speakers over there, and then we have the great dinner together. After a few days of time here with my family, I will fly alone to San Diego to stop by JCVI, my new institute, and then to embark on my 2 weeks nonstop trip again to San Francisco, Denmark, and Sweden to give 6 invited talks until September 2nd. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Leonard. He does not need any introduction because we all know him. He is the Charles Deering McCormick Professor of Teaching Excellence at Northwestern University and also the CSO of SynBYSS, a company. I have known him for more than a decade, and he kindly invited me to give a seminar at Northwestern University before I got tenured. And you know how important the pre-tenure seminar is for an assistant professor, and he kindly invited me at the time. He has been very active to promote science and engineering, especially NIH-related research, and we thank him for his scientific contributions and his service to the community, including the leadership at EBRC, Engineering Biology Research Consortium, where we both serve as a council member. I said I introduced him briefly, but that's probably impossible. Josh, thanks again for your contribution to nurture young people and also do great service to the community, and also for your great science to the community. And the virtual podium is all yours now. And thank you again.
Great. Thank you, Tasek. So let me get this up. Great. Can you see this okay?
Yes, very popular.
Well, I'll reciprocate as a start. So thanks very much for the invitation to join you at SynBYSS today, Tasek. I've been a longtime fan of the series, really impressed with the spirit and sort of the engagement this has achieved with reaching people and really in the spirit of SynBYSS. I'm just going to say a couple of words that I think might be useful, especially for folks who are researchers that are in or considering a career in the area of synthetic biology. And I'll make a few comments about just my own career trajectory, so folks on the webinar have a sense of where I'm coming from. A few thoughts, maybe some hot takes on SynBio as a field, specifically as it pertains to medicine. And I'll wrap up with a few lessons that are geared at sort of what I think is most useful in retrospect for thinking about, especially as one is carving out a career in this space. So say a few things on each of those bits there. And I will say in advance, I'm not going to talk about any details, but I'm very happy to connect by LinkedIn, talk to folks offline afterwards too. So if you're curious, please just hit me up. Okay, so a little geographic background of where it came through. And one thing that I do at Northwestern here is I run a biotechnology training program. When we have speakers come in, we ask folks to do what I'm going to do right now, which is talk about their own career trajectory. And I think it's surprising just how wild each one of these ends up being. So I'll tell you about my version of this. I grew up in the desert of Arizona, and that's where I really got interested in being an engineer in the first place, because I became interested in sustainable energy from middle school, basically. Started working in the solar space kind of in projects and eventually some internships through there. That's why I decided I wanted to be an engineer in the long run. Before going to college, I had a chance to live in Germany for a while in an exchange program, which was beautiful. This is a picture of Regensburg, fantastic city. Got me really interested in figuring out how to do a job that was both impactful in the world and maybe engages people well beyond the borders of where you're situated. Did my undergraduate at Stanford University, where I studied chemical engineering. During that time, worked at a number of jobs in energy and sustainability, including a stint in LA, where I worked for Southern California Edison, really trying to make a lot of big commercial units there more energy efficient, and eventually working at, during college, a company called SynBYSS, which doesn't exist anymore, but really pioneered the area of automated high throughput experimentation in that period of time. I got really interested, as I said, in energy and sustainability, but it became very clear over this period that one of the major barriers then and now was technological, but the other major barrier was policy. And so to really dig into that, I spent a summer working in Washington, D.C., where I was with the American Institute of Chemical Engineers, working to try to address a policy and really learn about how science and policy come together to make impact in the world. We were part of a project that eventually helped to spur the foundation of the National Institutes of Biomedical Imaging and Bioengineering, NIBIB. So it was really fun seeing how the sausage is made in DC. And again, informed what I wanted to do as my career too. So putting all that together, I became interested in sort of how do you merge science and impact and technology. I was very interested in the commercial and research side. So I decided to do a PhD. I did that at UC Berkeley, where I studied with Dave Schaefer, one of the pioneers in the field of gene therapy. That was a pivot topically, but really met the same idea of thinking about how to take science and make an impact in the world. And I really saw the emergence of bioengineering and gene therapy as one of those places that was ripe for contributions where the work you do day to day can make an impact. After studying with Dave there in gene therapy, I decided to carve out my own path. And I should say, during the time I was at Berkeley, I did a handful of classes in the business school and got interested in kind of how academia and business can work together. And that's really how I made the hard choice of going down the academic route. I'll come back to that commercial connection that Tae-Suk mentioned a little bit here. But I decided to find a new area that wasn't really connected currently to what I was working on. I wanted to learn about immune engineering. And so I did a postdoc in immunology at the NIH in Bethesda, Maryland, and then put all that together to pitch to Northwestern University, where I am now, the idea that we are at the point where we can use engineering to do more things in biology. And anybody at this webinar will recognize that as the pitch for synthetic biology. And at the time, there wasn't a ton of synthetic biology in medicine. That was really where we tried to carve out a niche there. And so I'll talk a little bit about those themes there, but I'll mention it does result in more recently taking that over the fence from academia to industry at our startup company, Cyanix, and I'll talk a little bit about how those pieces fit together here. So without any synthetic biology talk, I think you're obliged to give your pitch on what is synthetic biology. And I'm not big on definitions, but I am big on sort of the shared mission that I think we all have here, which is really to make the engineering of biology engineering, right? It's to make it so that you can start from coming up with a wish statement and have both the knowledge and processes and tools to actually build, prototype, refine, and ultimately have that work in a way that looks like real engineering. And sort of how that looks evolves over time. But from a medical perspective, why does that matter? The building takes the majority of the time in a lot of these projects. And so if you can make the engineering faster, you could spend more idea evaluating good ideas instead of building the thing that lets you evaluate the good idea, right? And that way make the development of drugs faster, address more needs, ultimately drive down costs of development and timelines. So that's the mission I think we need to all hold ourselves to every day as far as what we're trying to contribute to the field here. And so in the early days, we had all these heady notions that if we could just engineer cells like robots, we could make them do things like sense drugs and seek out disease and kill disease cells and program them in to be safe and effective. And one of the really cool things is that by and large, that promise has started to be realized in terms of medicines that are benefiting people in the clinic. There's a lot of ways to go here, but I think it's been fun to see over the course of my career, this go from an abstract notion to beginning to be a reality that impacts real people's lives. And so why is that? In a lot of ways, I think it's because you can say that mammalian SynBio has approached what we would recognize as more of an engineering discipline. So what has enabled that? I think it's not just doing proof of principle experiments in the lab, but also thinking about what is the knowledge we need to make that work all the way into the clinic. How do we not ignore the fact that all the cells are different and all the people are different, but build things so that they work in the context of that variation? And a really emerging theme right now is thinking about the fact that all this, we're using cells and the biology of our bodies as a therapeutic. That means that it has to carry out its natural activity, maybe controlled in a certain way, but we need to do all these interventions in a way that doesn't overburden the cells. Make sure that this works well, make sure that they're safe in a practical context. And that's where the challenges are. Not can we make a thing do a thing? We've kind of got to the point where we can do that. It's really now how do you address those things that are pragmatically well aligned with the translation at the clinic. And so, although it's certainly a lot of room to go, I just came back from the mammalian synthetic biology workshop last year, which is sort of the nucleus of the mammalian synthetic biology community. I think it's fair to say that we have a lot of different ways now of building new functions, using everything from kind of intuition to increasingly a lot of good computational tools, classical modeling based upon differential equations, which are just describing how concentrations change in time, to up to and including advanced methodologies of machine learning and AI. And in a lot of ways, they're doing similar things in the sense that it helps us get from a goal to a design. And increasingly, this is something that I think we can claim we're doing in an engineering type way. One of the exciting and I think sobering aspects of this, though, and this is a comment made by a number of folks in recent years, is that we still have a big way to go to making this benefit patients. And so here's my little summary of kind of why that is. If you look at the advent of the Human Genome Project, in the 20 years since the Genome Project was completed, this has gone a long way to helping us understand the drivers of a disease and even to come up with ideas as to how we could tweak biology to take those drivers of a disease and treat the patient using that biological information. The advent of technologies, including SynBio, but of course also things like CRISPR and other types of technologies, these are all great new ways of being precise and kind of having molecular level interventions that could be used to harness that knowledge. One of the big drivers in places where everyone's working right now is the delivery challenge. How do we take that thing and actually make it work in a patient's cells in a safe, cost-effective, and accessible fashion? Big challenge in the area here. And so I'll just end with one little bit that this is what we got interested in about 7 years ago now, looking at ways of solving this using biology. And I'll just mention, if you haven't read this book, a big pitch to read The Structure of Scientific Revolutions, because this is a good way of kind of understanding when you're in the process of upheaval in the way people think things work. And that's a huge chance to jump in and identify an opportunity to solve a problem that's been persistent over time. And so we jumped in at trying to understand how extracellular vesicles, or EVs, work. These are little particles the size of a virus. We've known they've existed since the 50s. And the conception has evolved from they are trash to potentially, in the recent years, they are the natural modes of gene delivery and other types of biomolecule delivery that could be used as a therapeutic if we could just figure out how to use them as a natural therapeutic. And so that's challenging, but we jumped into this recognizing that a lot of the tools we have for virus engineering and for gene therapy engineering could be deployed for harnessing this new category of particles. And since I won't show you the results today, I'll just say that both in my lab and as a field, in the past, say, 7 years especially, there now are a lot of great tools for being able to actually engineer these self-assembled particles to do things. This piece of artwork is representing a paper we recently published showing we could deliver CRISPR to primary human T cells and do gene editing to knock out the HIV co-receptor, kind of just showing that this technology has matured to the point where you can use biologically encoded drugs in a way that are very sophisticated and can do a series of sophisticated things that are ultimately well positioned for manufacturing. So the last point I'll mention is that this is kind of why we spun out the company. Cyanx is a company focused on making innovation accessible. Really what we're trying to do is solve delivery for everybody who's got a great new technology and really wants to figure out how to deliver that to a patient in a way that is cost effective, fast, safe, and efficient. And so that's our role in the world, is helping other innovators to do this. And it's been really satisfying. We actually, the call right after this is going to be focusing on our first product, which is hopefully going to be in patients either late this year or early next year. And so that'll be someone with hopefully a better outcome for cancer because of a technology that we've developed through the company and through this process. So I will end on just a couple of comments here before I hand it over to Rock here, which I think is just really useful for thinking about how, like, this is my take, but I think a shared take really for folks who've kind of gone down similar roads. Everyone's career is nonlinear. Everything sounds like a really good story at the end, and it never feels like that in real time. It always feels like a series of impossible choices. And one of the important lessons is there really is almost never one right answer. If you think it through and make your best guess, most people are happy with whatever they chose at that point, which means there must be more than one right answer because no one's perfect at guessing the right answer, right? The other real trick is you need to learn how, as a researcher, to encumber risk intentionally. I wish I could say this in a thousand ways. It's not, risk is not bad, risk is not good. You need to be conscious how much risk you're taking on and take it on in a way that balances things. So avoid the perils of playing it safe or risking it all. And especially as you're a graduate student where it feels like the outcome of your project is the outcome of your career, it's not. Your project is not your career. Your project has risk. And if you treat it this way, then you can sort of manage that risk in a way that works for you and ultimately lets you be successful in the long haul, because that's how science really works. Changing direction is good and often necessary. This usually, you can certainly drop things that aren't working. The harder things to do is drop things that are working because you're choosing to focus on what you think is more impactful. So learning to do that and let go strategically is one of the most important lessons, especially if you're moving into a leadership position in academia or industry. Very few things in emerging science are fixed. This is just the idea that everything seems like it's solved, but basically nothing is. And so when you want to jump in, don't worry about rocking the boat. If it seems like you've got an idea or finding that might disrupt the status quo, it's worth pursuing because many, many things can be disrupted in a positive way. And finally, I think the biggest piece of advice is choose to spend your time on work that makes a difference. Do something that you want to spend time doing. Do something that will make things better, and you'll always be happy to do the work that you're doing. So that's a luxury with this kind of a career. Important to recognize that, but something that is achievable on all kinds of tracks within the research sector and just kind of a useful true north to keep in mind. So stop there, hand it over to our main speaker today, Rock, who I know will manifest many of these things that I just talked about right there, and I'm very excited to hear your talk today. Thanks, Tisuk.
Josh, amazing. I mean, I love it. All the lesson or the advice you giving. That is so lovely. And then I believe you are doing so by doing multiple things, including your company. Because not many engineering professor, I believe we should do that, but having company and then really translate the technology into the real field, because I believe it is time for us to deliver to benefit the people. And then I absolutely excited because you're talking about cancer, because actually my dad also currently suffering from stage 3 cancer, but I hope your technology really available soon. In that way, many people, many family members benefit from your technology. Thank you so much.
Thank you.
And also, this is a coincidence. You're talking about, now I learn how to pronounce the area, Germany, Regensburg.
Yeah.
I going there in September for a seminar or the conference. And then now I know how to pronounce that one. I used to pronounce Regensburg, but that's wrong pronunciation now. And then Dave Schaefer coming to San Diego, I just saw in the morning whether I should attend that conference, bioengineering, whatever conference, but he's a keynote speaker. So, all right. Okay, thank you so much again. So let's go to the next session. Okay, now the main speaker of today with a longer introduction. Professor Zhi-Yan, or we call him Rock, Professor Rock Du, received PhD from the University of Hong Kong, working on lipid metabolism in plant development and stress response. After that, he did postdoc research at Michigan State University, focusing on microalgal research for lipid metabolism, cell development, photosynthesis, stress resilience and tolerance, and variable bioproduct production, and SynBYSS. In August 2020, he started his lab at the Department of Molecular Bioscience, Bio and Bioengineering, University of Hawaii at Manoa. His current research interests includes synthetic biology in microalgae for valuable bioproducts, symbiosis between microalgae and fungi, lipid metabolism in photosynthetic organisms, and improving traits of tropical crops. Importantly, he has been helping me as a SynBYSS conference committee member for a while, including booking the venue at his university conference hall and initial partnership between his university and J. Craig Venter Institute. He's truly a rising star with multiple funding and exciting research, and he is already being considered to prepare for his tenure package even after 4 years of his time or experience as an assistant professor in the university, including, of course, the pandemic period. So that is truly amazing. Rob, thank you very much for your time today, especially at the very inconvenient early time in Hawaii. I believe now Hawaii probably 5 a.m. something. And then please take it away.
Thank you so much. That's a great introduction. Appreciate that. And a great talk from Josh. I'll share my, let me share my talk. Okay. Well, thank you so much. Should I keep the video panel or?
No, that looks fine.
This is good, right?
That looks very good. Thank you.
Great. Oh, thanks again for the introduction and the opportunity for me to give this talk. Very exciting opportunity again. And today I want to explain the research I have right now on algae, fungi, SynBYSS, and the applications with synthetic biology. So I started in the University of Hawaii at Manoa 4 years now. So talking about Hawaii, this is where I am. We're right here on the Oahu Island. Well, when you look at this map, it's kind of scary because you're in the middle of nowhere in the Pacific Ocean. Plus, we have lots of people here, like over 1.5 million residents. And just in Oahu, we have over 1 million people. You see how big the island is. We don't have a lot of land, lots of resources. But it's kind of a paradise, very nice view. This is Manoa campus, and this is where my lab is, the Agricultural Sciences Building. We started in August 2020, right during pandemic. And that's the view from the other side. We're right into the Manoa Valley, beautiful city. And I'm in the Department of Molecular Biosciences and Bioengineering. We work on the fundamental research and applications for agriculture and aquaculture. my college is tropical agriculture and human resources. Agriculture is a very important topic for us. So as a new faculty, my priority is to set up the lab. I've been doing that for many years now. So luckily we set up the lab with the essential equipment. Like you can see here, digital PCR, lots of microscopes. We have our own confocal and the digital microscope. We have our GCFID and other equipment for our research. And for my department, we have the proteomics and metabolomics core facility, where we have more mass spec, HPLC, and other equipment, plate reader, the autosomal titration calorimeter. So these are important for our research. And we just got 2 new mass spec from Agilent, 1 LC mass spec and 1 GC mass spec. So with these resources, hopefully we can make more progress in our research and education. So for my lab, currently we have several different projects. I work on both algae and fungi. For today's talk, I will explain our research in synthetic biology, microalgae, and the symbiosis between algae and fungi to improve the performance of algae and solutions for some challenges in industry. Here are my current graduate students. We can't make progress without their efforts. And Michelle, she just finished her visit and lab work. We're working together on several research papers, and hopefully we get some more funding to invite her back. We're always looking for new PhD and postdocs. If you're interested, please go visit our lab website and you can find more information about our research. From the introduction, I did my postdoc research at Michigan State University with Dr. Christoph Benning, where I started working on microalgae. Very nice university, nice research building. And I have many collaborators there to support today's talk. And I started collaboration with Dr. Towan and Dr. Christine Parent last year. So hopefully we can do more work into the mechanism about the research topics we're interested with, like with solid state NMR and cryo-EM. So I want to thank them for the support. And here from this photo, you can see there is a challenge for me when I was in Michigan. It's the long winter. It's very cold. For my research, it's not a good thing. Back to Hawaii. So we're in the ocean, kind of scary, but also we have lots of advantages. A big advantage here is the year-round climate for agriculture. So we can grow crops, algae year-round. Like here from Bayer, Hawaii, the breeding and the seed production is very important, and we can do this year-round. For aquaculture, like here on the Big Island, the Kona neighborhood, we have lots of aquaculture and algae companies. In Hawaii, over 50% of aquaculture is from the algae industry. Here, the warm temperature and rich resource of fresh and salt water, low pollution is ideal for the industry. Here are some algae companies and my collaborators in Hawaii, like Sanotech, Sambosha, Oceanera, Conley, Ergosystems. So work with the industry partners here. We want to translate our findings for actual applications and also support the local communities. Using microalgae, we can make lots of products with their very useful biomass. As you can see here, they have lots of good lipids, proteins, carbohydrates. We can use them for nutraceuticals and a lot more. So for today's talk, I'll explain how to engineer microalgae for high-value bioproducts, and we developed 2 solutions for the industry challenges. One is to harvest microalgae and how to increase their density. So first topic, engineer microalgae for bioproducts. We use Nanocropsis oceanica as the model system. So this is a marine algae, has been used for lots of industry products because they have very high oil and EPA. They're very robust and resilient with a small genome, which is very friendly for genome editing. In my previous lab at Michigan State, we developed lots of genetic engineering tools. We have the genome data. We also have a marker-free CRISPR system using the episomal CRISPR method. So these are the resources we have to support this research. Using microalgae as a platform, we want to produce useful compounds. For example, plant terpenes. I work with Dr. Bjorn Hamburger at Michigan State, who's the master of plant terpene research. In the lab, we have different terpenes, and we were interested to explore this class of compounds. It's a very large and diverse family. Like, for example, in cannabis, there are over 120 identified terpenes. They have very different applications, for example, as azancing, drugs for cancers, perfume, flavonoids. So in the lab, we use plants as the resources. They have lots of different new pathways, genes, and compounds for us to explore, and we can get the new resources for more bioproduct engineering. For plants, they're a good resource for the new pathways and genes, but they're not the ideal production system. So here I want to use microalgae as the superfactories because they have the unique advantages. Similar to plants, they have chloroplasts. They can do photosynthesis, and they have lots of precursors available. And they also have advantages like microorganisms. So we can combine these advantages into a super system for bioproductions. So for this work, we select one of the diterpenes, caspine, as the model system. Caspine is a diterpene, can be used as precursor for many different terpenes with different applications. Again, in the Nanocropsis algae, they already have chloroplasts and the MEP pathway similar to the plant host. But they don't have the MWA pathway in the cytosol, missing several key enzymes. So with this system, we want to use synthetic biology tools to reestablish the MEP pathway for caspine production and use key enzymes like the DXS, GGPPS in the pathway to increase the production of caspine. So to start, we develop constructs. First, we want to focus the engineering in the chloroplast. So we have constructs using the plastic targeting peptide right here, and localize the foreign genes like the terpene synthesis one into the chloroplast of the algae, which is right here. in the green fluorescent. The red colors from the chloroplast, and this is overlap. So we're able to locate our genes into the chloroplast, and we're able to produce caspin using the caspin SynBYSS. So this is a preliminary test. We're able to produce caspin in the algae. This is a diterpene with antibacterial and antifungal activities, and it's a precursor for many different terpenes. So in order to improve its production, we develop more constructs with the gene editing tools we found from the algae's genome. And we have the P2A, for example, the self-cleaving peptide, so we can put multiple genes in one construct, and we have the rebi directional promoters. It's a strong promoter and can drive the genes from two directions. And we have the NLUX, which is a nanoluciferase for mutant screening. To improve the production of caspin, we have the two key enzymes. In addition to the final caspin SynBYSS, we have the DXP SynBYSS and the GGPP SynBYSS, using the P2A to combine them together. And we also have the single caspin SynBYSS as control. So this is the overview of the procedure. We will perform the transformation using electroporation, go through the screening process, and then GC mass spec and biomass analysis. One important method here is the screening process. With the nanoluciferase, we can do a high throughput luminescence screening. The method is like this. We use 96 wall plates. It's like colony PCR. We can see positive, negative transformants from the screening. Plus, from the nanoluciferase, we can select high expression strings. The result is the signal is very strong, so we can put a threshold and select the highly expressed strings for further mass spec analysis. This is the final result. By putting the two key enzymes to increase the precursors for the final caspin production, we were able to achieve much higher yield for caspin compared to the solo terpene SynBYSS control and the terpene SynBYSS fused with GFP. So this is kind of a proof of concept. We're able to engineer microalgae like nanocropsis to produce plant terpenes. And there's a big treasure for us to explore, and the future work is to test more different terpenes from different plants. So now we see the potential for microalgae in high-value bioproducts, but for microalgae themselves, they also have some issues, like the two challenges here. You can see the culture, the cells right here. This is the nanocropsis culture. They have this low density challenge because they're very small, and they're in the culture. It's very expensive to harvest the algae. In the lab, for example, we can use centrifuge to harvest microalgae. I found this video online. This is more like a special centrifuge developed for algae harvesting, but we're talking about industry scale, like the big open ponds here. No way we can use this size of instruments for the commercial products. So Instead of using $1 million of facilities for harvesting microalgae, we want to develop some solutions. Like, for example, I remember when I first started in the lab, sometimes we got contaminations in the algae culture. It's very frustrating, so lots of effort wasted. Normally, you autoclave the materials and start all over again. But from this observation, the fungal, especially for fungal contamination, they can flocculate microalgae and form some aggregates at the bottom of the flask. So, inspired by that, we want to try whether we can use fungi to harvest microalgae. I collaborated with Dr. Greg Benito, who's the expert of fungal research and mushrooms like truffles. So we started this test of algae and fungi interaction. So this is the summary of the result. We tested many different strains, summarize the findings. Like here, that's the strong interaction, medium interaction, and no interaction, and also algae alive or dead. From the photos here, you can see there's a diverse response to this fungal interaction, which means lots of interesting mechanisms for us to explore. And among all the fungal strains, we were lucky to identify this family, Mozzarella fungi. They can flocculate nanocropsis very well. So this family of fungi, they're widespread soil fungi. They're not pathogens, and they've been used in industry for many years, similar to some of the microalgae, because they can produce very high amount of oil. This is a fungal hyphae. You can see the lipid droplets inside the fungal hyphae, and they have very high amount of arachidonic acid. For example, from the Mozzarella alpina, they can have up to 70% of arachidonic acid. So don't be surprised when you look at the ingredient list, you will see them from lots of products on the market. So we can flocculate microalgae like nanocropsis using Mozzarella fungi, and we try some other fungi, use the strains, for example, Mozzarella americana here as an active control, which is very sad at that time because we really wanted to work. And this is the mycelium of the morel mushrooms. So it's the efficiency is efficient, not the best, but they can flocculate microalgae like you can see here, and no obvious morphology change for the algae. Plus, we can yield very healthy fatty acids from this combination. This is a confocal result. Those red colors, those fluorescent signals indicate lipid droplets in the algae and the fungi. And this is the fatty acid profiling. They have lots of ARA, EPA, and DHA. In addition to nanocropsis, we tested different algae, like green algae, Chlamydomonas, chlorella. Their efficiency is much higher. And we also use alpina here as the partner to form the aggregates. So the challenge again, how can we collect the aggregates from the co-culture? Again, we're talking about industry applications. That's a different level of biomass. We don't want to use fishing net or something to pick up the aggregates. So we developed this system. It's a very simple design. That's the first generation of this harvesting system. We use the fungal mycelium, we stack them together, and we have algae culture go through the mycelium. And as you can see here, they can collect the algae pretty efficient. This is before the filtration, this is after. We were able to reach over 90% of harvesting efficiency. Plus, the fungal mycelium, they're kind of like filters, but they don't collect algae as filters, which I will explain in the next section. After the harvesting, we can dry the aggregate. They look like seaweed, and I did try this for my fish, and they love it. So give us some potential for use this biomass for more applications like for food, nutrients, and feed. So when I started in Hawaii, we have more 3D printers, and I have a team of students trying to develop more systems with more prototypes. We want to work with the industry to develop the commercial scale harvesting device. Well, because of my tech transfer office told me I can't show too much details about this, so just some images. I'll skip the videos here, but we're trying to develop the prototypes for industry to harvest microalgae. So, well, last section today is how to improve the low density issue. Quick recap, we have the screen aggregates from the algae and the fungi interaction. In order to improve the interaction and increase the density of the algae, we want to look into the mechanism, how algae and the fungi, they interact with each other, or how fungi can capture the microalgae. This is a video showing the process. So the fungal mycelium, they can trap the microalgae in the culture, pretty efficient. And we did many microscopy work, like for example, the SEM research here. We see more details, how the algae can interact with fungi. This structure reminds us Velcro tape. It's a very interesting cell wall interaction. What make it more interesting is for the algae, they do have a smooth coat over those fiber-like extensions. So we see the broken cell wall from some algae during the interaction. So we did more analysis on this. In the clean algae culture, they do have the smooth surface. And after the interaction and co-culturing with the fungi, we can release those fiber extensions, which we won't see in the samples without physical contact between the algae and fungi, and if the fungi, if they're dead. So we can partially mimic this process using the combination of dressylase and hemicellulase. So we can release the extensions. Again, we don't see this kind of phenotypes in the negative control in the Morel Machella americana. So this findings is consistent with the previous cryo-EM result. You can see this coat covering those extensions. So we want to do more work to understand the mechanism here, why the algae do this, what is the regulation here, working with my collaborators at Michigan State, Dr. Chowang, with the solid state NMR, and Dr. Christine Parent using cryo-EM. Hopefully we can dig into the mechanism for this observation. And get back to the structure here, if we zoom in to this the tubing-like structures, it feels like there might be some nutrient exchange. To test that, we developed this labeling experiments using C14, sodium bicarbonate for the algae, C14 glucose and acetate for the fungi, so we can label either the algae or the fungi and then track the exchange between the two organisms. Here's the result. So we divide the compounds into 5 groups: lipids, free amino acids, proteins, soluble compounds, and carbohydrates. So from the algae and the fungi interaction, this is the result. C14 labeled algae, and we did see C14 in the fungi, and a lot of them in the lipids. For the labeled fungi to algae, we also see lots of C14 in the algae. What is interesting here, it's for both algae free in the culture and for the algae attached to the fungi. A lot of the signal is in the lipid. For the attached algae, also in proteins and carbohydrates. So this makes us want to investigate more, like, is the physical contact necessary? To answer that question, we design more experiments like this using small inserts. This insert has this membrane bottom, which allows metabolite exchange, but they can physically separate algae and fungi. This is what they look like when they're in the plates. To confirm that we don't have any cross contaminations, I have the two set up right here. Like, I have the algae in the insert and fungi in the plate well, and then opposite, algae in the well and fungi in the insert. You can separate them very easily. Just transfer the insert to a clean well, and there's no cross contamination. This is a view from the side. So very simple but very efficient method to test whether the physical contact is necessary. And in summary, this is a result. For algae to fungi, the physical contact is it is necessary. Without the physical contact, C14 is very low. Same for dead algae. So the algae needs to be alive, and they do need interaction with the fungi for the fungi to receive the C14. For algae, it's a different story. They're not very picky. We can detect C14 with or without physical connection, live and dead fungi. So that's a summary for the carbon exchange, and we did similar experiments for nitrogen, which is very important for microalgae. And we see the nitrogen exchange between algae and the fungi, which is not a surprise because we actually see different mozzarella fungi to release organic carbon and nitrogen into the culture. So with the nutrient exchange, fungi can support algae for better performance, especially under stress. We challenge the algae with nitrogen starvation and use the settle screen to investigate the viability of the cells, and we see a significant improvement when algae is with fungi. For fungi, we did biomass analysis, and we see the higher biomass in the algae fungi aggregates. So it's a mutualism with nutrient exchange and support from each other. So this gives us good confidence, so we can use the method for the low density issue. For the short term cultivation, it worked, and we also tried the long-term cultivation. This is a co-culture for over a month, and we see some very interesting phenotype. The fungi, they are alive, algae, they are alive, plus with the confocal microscope, we see some interesting phenotype. Some microalgae seem like inside the fungal hyphae. To confirm that, we did lots of TM screening, and we found lots of cross sections like this. We have the microalgae structure inside the fungal hyphae, and we have a healthy structure of chloroplasts, indicating the algae, they're alive. And in fact, the best method to study this phenotype is light microscope. So we observe lots of green hyphae with microalgae inside the fungal hyphae, forming photosynthetic mycelium like this. And I have a video here. It's a time-lapse video with images taken every 10 minutes for 6 days. And inside this fungal hyphae, we see this group of microalgae, they're dividing. And both algae and fungi, they're not dead. We use some dead cell controls, and we don't see that in this. So with this system, we can combine algae and fungi together so we can condense the biomass of algae. So this can increase the density of the algae culture and make it more productive and save space and other resources. So this is a summary for today's talk. We developed the microalgae system to produce plant terpenes using synthetic biology tools and methods. And we developed 2 solutions for the challenges in algae industry for harvesting and increase the density by co-cultivation. I want to thank my team and funding resources, and I'm ready for questions. Thank you very much.
Thank you. This is amazing, Tom. I love it.
Thank you so
Actually,
much.
I have question already from Josh, but Josh need to go for another meeting in 11. So he asked question and then he actually delivered that to me. So let me ask you the question, Josh actually asked. So question is, to what extent does the circadian cycling of sunlight when deployed in outdoor pools impact metabolic engineering of microalgae? That's first question.
Sorry, can you say it again? I
So
missed.
to what extent does
Uh,
the circadian cycling impact metabolic engineering of microalgae?
That's a good question. I don't have a solid answer now. What's the next question? The second
The
question?
next
I
question
can get
is
back to the first question later. Yeah.
probably, yeah, so next question probably, I don't know, related or not, but do you see the need for different genetic engineering or control strategy for use in continuous bioreactors versus outdoor ponds?
Yeah, they're sorry, can you explain the question again?
So do you see the need for
Mhm.
the different genetic engineering or control strategies for use in continuous bioreactors versus outdoor pond? So I guess his question is there are multiple ways to culture your algae, micro algae. One way is a continuous bioreactors, and then that's a control, very control, in the bioreactor. And second option would be outdoor ponds. Uh,
Mm.
so and then he asking, I believe, do you see the need or different genetic engineering is necessary or different control strategy is necessary when it comes to continuous bioreactor versus outdoor ponds? It's a little
It
bit,
sounds like there are a couple questions for that question.
yeah,
So first,
together.
it's because when we do outdoor,
Uh,
there are lots of regulations for genetic engineering. So we
right.
don't have GMO
Yeah.
materials has the chance to release to the environment. We're in Hawaii, it's a very fragile system, so we want to be very careful about
Right.
that. So I showed some potential collaborations with industry in the future using the GMO, possibly GMO strains. But if we go that way, it needs to be by the non-transgenic marker-free CRISPR. So we
Yeah.
can develop the marker-free CRISPR system. Basically, it's an episomal CRISPR method. After the Cas9 editing, the mutation introduction, then we will remove the selection pressure, and the algae will degrade the episomal plasmid.
Yep.
Then will be clean. They keep the mutations, but they will be clean.
So you are
Uh,
saying the mutation is
mm,
okay, but introduction of heterologous genetic part into the
mhm.
microalgae is not good for the regulation, right?
Yeah, at least for the industry, yes. I don't think it's ready for that.
Yeah.
Um, for that, we probably need to go for different approaches. Um, I also have some mutagenesis breeding ideas, and that's kind of my priority now because
Mm-hmm.
I want to develop the non-GMO strains for the industry. So,
I guess that's the question he wanted to ask, and because that is basically the
mhm,
concern when it comes to GMO. Because if you
yes,
introduce some new gene from another organism to microalgae, that's become GMO, and then
yeah,
heavy regulation. Uh, I mean, mainland, a little bit generous, but Hawaii, I heard, very, very restricted because of the unique environment over there, right?
yes, yes. I think it's for everywhere. It's just because here, our regulation for the general materials. I'm not talking about the GMO materials. I'm talking about, like, for example, if I want to import some wild type algae or fungi, we need to go through the regulation process
Mm-hmm.
with and quarantine office from the Hawaii
Yep.
Department of Agriculture. So we have a very critical process how to do that. So for the cultivation system, outdoor system apparently has like a different level of
Mm-hmm.
regulation compared to the lab research.
Yep.
Use
Absolutely.
the more controlled photobioreactors in the lab, we're able to test both GMO and non-GMO algae and fungi. And yes, that's it's for research, and also eventually it depends on what it's for. Uh,
Mm-hmm.
like for example, if the final product is for food, feed, some nutraceuticals on the market, we don't really need to do the genetic editing to
Mm-hmm.
produce the plant terpenes.
Mm-hmm.
Those compounds are like high value
Mm-hmm.
products.
Yep.
So with that, we can afford to have more expensive setup using the photobioreactors
Mm-hmm.
indoor and manage the GMO materials.
I
Uh,
see.
yeah, well, in Just Talk, he he's showing a great example of how to combine research and academy research and industry.
I see.
So yeah, here this is also what we want to do. It depends on the project and the material. If we are aiming for like a super factory using microbiology to produce drugs, for example, then we can use the indoor system bioreactors, the fermentation system to produce the compounds using the GMO strains. But for the outdoor cultivation, is more for food products and
Got it.
food and nutrient products.
Got it. So yeah, that's kind of, I guess we answered his question. So that's more likely, the difference between when it comes to indoor or contained bioreactor, and then that issue is the GMO, kill switch, blah, blah, blah. So we
Yeah.
need to technologically implement or do some different approach to make that happen in the different environment. And then the first question is, I believe this is a challenging question, but he asked, I put in the chat, to what extent does the circadian cycling impact metabolic engineering of microalgae? Because sunlight, changing outside, I guess that's kind of what, because he talking about still outdoor situation. If you kind of put the algae in the open pond, you have the cycling of sun and the dark, and then in that way you may need to do some, I don't know, you may
It's
have some
huge.
problem of circadian cycling problem when it comes to producing something or engineering something.
Yeah, so yeah, now I see the question better. So for this is a great question because this is actually a very important challenge for industry. And also one of the reasons why Hawaii is very attractive for algae industry. Like I mentioned, the weather here is nice. So it's not just warm temperature, it's reliable. It's very stable. So they don't change much year round. For algae, they don't really like strong sunlight. That can kill the algae. So that's why, like for example, in my slides, I show the photos from the algae farms on the Big Island. Like in industry, they actually use the seawater not just as nutrients. They also use the seawater as a cooler, so they can cool the algae. Like I said, the weather here is stable. Sunlight, it won't change that much like on the mainland. And it helps algae to be stable. And plus, they don't have, for algae they do this in cycles, and not very long cycles. Microalgae, they're kind of like well, not as fast as bacteria and yeast, but they're also robust. You don't have to grow them. It's like batches, right? So you can harvest them when they're ready.
I see.
Yeah. So the circadian, yes, it's a challenge, especially when it's very hot. When the sunlight is strong, but again, it's in Hawaii. I can't speak for that on the mainland. Apparently, that's a challenge, right?
Sure.
You have bigger changes on the mainland.
Sure.
But here is more stable. Temperature is a bigger issue.
I see.
Compared to sunlight.
Okay.
In the lab, we can actually grow the algae under continuous light. 24 hours, 24/7. Yeah.
I guess the question is about that one. So in the
Yes.
lab, you have the continuous light, 24 hours, and the outside, half of the days I mean, the day is basically the light, but the night is the dark, and then that changed probably something like physiology of the algae, and then the way you need to do engineering. So I believe the question is about that one. So going
Not
from the
necessary. Yeah, not necessary
not
actually. So
necessarily.
yeah, we can set up. I have incubators. I have shakers. We have timers. We have controls to turn on and off the light. So
Yeah, yeah,
we
yeah, yeah.
can.
Absolutely. So, but the question is, in that case, I mean, if you do the continuous
Mhm,
light situation and then dark and night cycle, like that
mhm,
dark and light cycle, so dark and day cycle, what is
mhm.
the difference in terms of productivity, in terms of the, other things? So that's the question, he asking.
Yeah, yeah. Yeah, well, actually, this is very important. When I did my postdoc at Michigan State, it's not my project, but we do have the circadian like control, the quiescence state of microalgae, especially in climate bonus. We need to synchronize the cells.
I see.
You if they're not on a different clock, you will see the difference.
I
And
see.
algae, they're um, is not very obvious from like from the cell density,
Mhm.
for example, but it's obvious when you look at their gene expression.
Got it.
Yeah.
Yeah,
So
absolutely.
we did lots of RNA sequencing from that. There are several papers published from my previous lab.
Absolutely.
Yeah.
Actually, my lab and Himadri Pakrasi lab also published multiple paper about the circadian, the
Yes.
rhythm, darkens, in life cycle that change entire, microbiome, no, the transcriptomic, change. I mean, basically that happened.
Yeah, it also depends on the algae strain. So
Right.
different algae species, they have like different levels of responses to the circadian regulation. So
Sure.
yes.
Okay, let me check whether we have the question from audience. I didn't see it last time when I checked it, so we don't have any question. And also, this is already 11:17, so we probably need to close and then chat, separately, without recording.
Yeah.
Okay, so let me close. So again, the fantastic talk. I really love it. Thank you all for joining and staying today. We'll meet again on August 22nd, Thursday, the same time, the same Zoom link. We'll have Professor Keith Tayo also at the Northwestern University, and then Professor Mark Mimi at University of Chicago. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I will stop recording. Just give me 1 second.